This video lesson will introduce us to the topic of cellular energetics and help us meet course objectives 1 and 4. Particularly, at the end of this lesson, you should be able to describe the structure and function of mitochondria. Mitochondria have a unique structure in that these intracellular organelles have two membranes surrounding it. This TEM image illustrates a typical mitochondrion. The size and shape of a mitochondrion depend upon the age of the organelle. They can vary in size from very small and round to very elongated. The number or density of mitochondria within a cell varies too, depending upon the particular needs a cell type has. For example, sperm cells have a single, large mitochondrion that is formed by the fusion of many mitochondria, while skeletal muscles will have upwards of 30% of the cell filled by mitochondria. When looking at an artistic view of mitochondria, we can determine the function of the different mitochondrial regions. These include an outer membrane, a fluid-filled intermembrane space, an inner membrane, and a fluid-filled chamber called the mitochondrial matrix. The outer membrane is in direct contact with the cytosol and has a 1-to-1 ratio of proteins to lipid, so about 50% each lipid and protein. The proteins of the outer membrane are used to transport solutes such as ATP, NAD, and coenzyme A in and out of the mitochondria, and some proteins act as enzymes. Pores are formed by porin proteins. Interestingly, the porins are not always open and can be closed upon certain conditions. The inner membrane has a greater proportion of proteins when compared to the outer membrane of a 3-to-1 ratio. This membrane does not have cholesterol, but instead has a molecule called cardiolipin. The inner membrane is very impermeable. However, solutes can be transported across it using specific transport proteins. Some examples of the transport proteins of the inner mitochondrial membrane include those of the glycerol phosphate shuttle, which helps electrons from cytosolic NADH get transferred to the mitochondrial matrix. Similarly, the malate aspartate shuttle can shuttle electrons from the cytosol to the matrix. Additional examples of inner mitochondrial membrane proteins include those of the electron transport chain and ATP synthase. Lastly, the mitochondrial matrix is a fluid-filled suspension of enzymes to power metabolic reactions such as pyruvate oxidation, also referred to as the intermediate step, Krebs cycle, and electron transport chain. The matrix also suspends mitochondrial DNA, RNA, and ribosomes. In summary, mitochondrial numbers vary between cell types, and mitochondria are intracellular organelles that have two membranes surrounding a fluid matrix.